Let's study about the flow cytometer. Let's analyze the word of flow cytometer. Cyto is a cell, meter is measuring, and flow is in flow. So we are measuring the cells in flow. So in this system, we make a flow, and then along those flow, we flow the cells. And by giving light, then we measure those properties. This flow cytometer has been applied to blood analysis, and in our blood, the majority of the cells are the red blood cells, so-called erythrocyte, and some small populations of white blood cells. Among white blood cells, there are some lymphocyte, monocytes, and macrophage, and granulocytes. And interestingly, if you go to hospital, they take our blood. In 10 minutes, they can tell how many red blood cells and how many of different white blood cells are in our blood. How can they do so fast analysis? That technique is a flow cytometer. If you irradiate light to the cell, now the cells can generate two scattering signals. One is a forward scattering and one is the side scattering. Depending on those scattering strengths, we can plot this way. And forward scattering gives us information about the size of those cells. Side scattering gives us the granule status of those cells. So more granule, more side scattering. Using this forward and side scattering, we can plot those blood cells. And utilizing this, let's say this is a small size and this is a bigger size. And this has low granule and this is a high granule. And from this, we can get an idea. This is a lymphocyte, T cell or B cell. And this is a monocyte like a macrophage. And this is a granulocyte. Red blood cells are much smaller than these white blood cells. They should be around here. But the dominating numbers of red blood cells can interfere with this analysis. Usually we do hemolysis first to remove all the red blood cells. This flow cytometer not only generates those scattering information, it can generate fluorescence information too. By adding fluorescence molecules to the cell, now it's possible to detect those fluorescence signals. In this case, this was green color. If you label with a red fluorescence, of course, you can detect red fluorescence. And this can be expanded to multicolor analysis utilizing different dichroimeter and multiple detectors. Combined together with this fluorescence, this flow cytometry can be called as a fluorescence activated cell sorter, abbreviated as FACS. Utilizing this FACS technique, not only just counting the cells, depending on those fluorescence signal, we can physically sort out those cells, green cells and red cells. We can isolate, and those cells are still alive. That means isolating out the cells in live status and reculture and utilization is possible. In flow cytometer, the scattering information can tell us whether this is a lymphocyte or the macrophage or the granulocyte. But inside the lymphocyte, whether this is T helper cell or T killer cell, we need another biomarkers utilizing these antibodies. Now let's say labeling antibody with a green color for TCD8 and CD4 for helper cell with the red color. Now we can label those cells, mixtures of cells with a different antibody, and then plot them. Then this is unstained status. We don't see anything with a green color or red color. This is CD8 and this is CD4. By analyzing those two colors, we can tell this is CD8 only, this is CD4 only, and this is CD8 and CD4, both express the cells. Now, if you have enough numbers of different antibodies and different color, it's possible to isolate and detect hundreds of different cell types using this flow cytometer technique. How about the cell cycle? In cell cycle, the biggest change inside the cell is the DNA contents. Do you remember? In G1 status, the DNA is the same amount ongoing, and then DNA synthesis stage, now the DNA is increasing. And G2, until it is dividing, the cells are double amount of DNA there, and then divide and back into one cell, 2 end stages. Now this can be monitored by this DNA binding dyes. Do you remember? Propidium iodide was a red dye, and they can enter only dead cells when the plasma membrane was broken and then bind to DNA and then generate strong red colors. So if you plot the cells along this propydium iodide intensity, now we can tell this one is around the 2N stage, this is a 4N stage. Then this population should be G1, and this is G2, and this should be S phase. Some cells may have a smaller DNA contents than 2N. Maybe this is the dying cells or the apoptosis cells. By this analysis, we can calculate those relative amount of G1 and G2 and S phase cell number and some of those dying cells. This is one example of a real situation. Now, utilizing this kind of cell cycle analysis, this is the G1 stages, usually highest population, and this is the G2M and S phase. Small populations of those cells are dying like this. This flow cytometer technique can be applied to apoptosis too. In healthy cells, in the membrane structure, phosphatidylserine is facing only inward. When the cells are going into the apoptosis, some phosphatidylserine becomes too exposed to the outside. And Annexin V is the detecting molecule for this phosphatidylserine. And this can be modified with a PC, green fluorescence molecule, and then it can show green colors outside of the cells. Even you can add propydium iodide, which can enter the cell only when the membrane is broken. Early stage of apoptosis, membrane is okay, only phosphatidylserine can be exposed to the outside. Later stage, membrane can be damaged and propydium iodide can enter. Then by analyzing these two colors, one is a green color and one is a red color. Now in the beginning, the cells are healthy. Now this channel is the Annexin V channel. Slowly the cells are going into the apoptosis. If it goes further, now membrane structure is also damaged. Now propydium iodide can enter. Now some cells has both Annexin V staining and propydium iodide staining together. So this is the late stage of apoptosis. Real example can be, this is healthy cells. By adding some toxic material to the cells are moving into the apoptosis and the later stage of apoptosis. As you can see here, using this fact sorting or flow cytometer, thousands of cells can be analyzed very quickly and it can generate a lot of statistically meaningful data. That's why this flow cytometer is a so powerful technique for analyzing cells. And unique fluorescence molecules combined together with the unique biomarkers, we have huge potentials to discriminate many different types of cells or their cell stages. Okay, this is the end of today's lecture. Now again, if you have general questions, and you can put up your questions to the IVLE. And have a good week and see you next week.